Ruparelia, and we have Ms Emily Galea. Three of them at the end, Meagan, Emily and Avnika, will be joining us to answer any questions that you might have. I have heard them speak before and it's absolutely fascinating. I hope you'll enjoy it and we will start the presentation. Thank you.
Right, hi everybody, my name is Emily and I am a research coordinator based at the Murdoch Children's Research Institute in Victoria, and we have got my colleagues Dr. Pete Houweling, Meagan McGrath and Avnika who will be also presenting today. So we're going to talk a little bit about our research into genetic muscle diseases. Next slide, please. So we are part of a group that's called a National Functional Diagnostics Program for Therapy Development into Congenital Muscle Disease. We've shortened it to ADAPT-CMD, and this is where we all started. So Professor Pete Curry, who is pictured there on the right-hand side, he leads a team of scientists and clinicians around the country, and he was successful in obtaining a Medical Research Future Funder grant for our work. Next slide, please. So the presentation today, we'll talk a little bit about the ADAPT-CMD and where it started, and the National Muscle Disease Bio-databank, which is the project that I coordinate. And then once samples go into the biobank, how scientists use them, and then working together collaboratively, and then we'll have some time for questions at the end. So I might hand over to Dr. Pete Houweling, who can talk about the team and how we all started. Thanks, Pete.
Great, thanks, Emily, and thanks everyone for the chance to come and speak with you today. So yeah, the ADAPT-CMD team is a group of scientists, laboratory-based scientists, as well as clinicians and researchers that are really working together to try to answer some tough questions about muscle disease. And so the ADAPT-CMD program is depicted over here on the right-hand side. And like I said, it's a real network of patient advocacy groups like yourselves in MD Queensland. You guys feature up here, and we're really grateful for the letter of support that you wrote for this program. It's integrating advocacy groups with the clinical networks that we mentioned down on the side here, and we'll touch on these clinical networks which are part of the NMDB, the National Muscle Disease Bio-databank, next. But feeding those networks into the laboratory, which is where myself and Enzo Perella at the MCRI, Pete Curry and Avnika at ARMY, and actually now at the Centre for Muscle Research, Professor Gordon Lynch and Paul Grigorovich, and Meagan McGrath and Christina Mitchell work together to really start to model the diseases and test new therapies. So this project actually leverages something that's already been established and that we hope that you're aware of, and it's the Australian Neuromuscular Disease Registry. And so this is a collection of clinical information from patients around Australia that have neuromuscular disease. And while this is a really excellent resource for the clinical teams and also for data gathering, it really was missing one crucial part, which is the patient material and samples that we need as part of the to do our research. And so that's really the first step of creating ADAPT-CMD, was to be able to develop a national framework to be able to collect these samples. And I'll hand over to Emily, who's going to tell you a little bit more about the network that we've created.
Thanks, Pete. So yes, as I said, my role is to coordinate the National Muscle Disease Bio-databank. And we obtained ethics in 2022 at the Royal Children's Hospital. So the biobank is housed at the Murdoch Children's Research Institute, which is on the same site as the hospital. And the biobank collects clinical data, tissue samples, blood, muscle and skin from patients across Australia that have a confirmed genetic muscle disease. And then this bank of data and samples are used specifically for research purposes. So when the ADAPT-CMD team was formed, it's very important to have that collaboration between the scientists and the clinicians. And we wanted to set something up nationally that we could recruit patients around the country and then bring their samples to the Murdoch Children's Research Institute for processing, storage, and then use for research. Next slide, please. So this picture just depicts the sites that are currently involved in the biobank. So we've got WA there to the left, and they actually have their own biobank because WA was a little bit far to get the samples here to Victoria, but we still work collaboratively with WA there on the left. And then we've set up being able to recruit at the Women and Children's Hospital in Adelaide, the Queensland Children's Hospital, the Sydney Children's Hospital and Monash Health, as well as the Royal Children's Hospital here in Victoria. So as I said earlier, the samples are collected at these sites and then brought to the Murdoch Children's Research Institute for processing. So how to get involved? So eligible patients can be referred to me by their doctors, or patients can make independent contact via the contact details there. And if that happens, we then ask your consent to confirm eligibility with your treating neurologist. And then from there, we can then go on to consent to the biobank if you are eligible. So this is the recruitment to date. As I said, we got ethics in 2022, and I will discuss this a little bit later, but it is a very lengthy process to sort of set everything up. And then in the last couple of years, these are the disease types that we have recruited along with the numbers. So a total of 19 patients to date. And then we've just broken up there the number of blood samples and muscle samples that we have collected in this project. So challenges of establishing Australia's first National Muscle Disease Biobank. So yeah, legal agreements between all the sites is very time-consuming process, as well as a site-specific approval. So once you sort of set up the main ethics, you then have to make sure that each site is able to accommodate the research, and that's a process, submitting documentation and getting all the sign off at all the different hospitals. Busy clinic workload, so we rely on clinicians to refer us eligible patients, and often they're very busy, and that can be a bit of a challenge. Logistics of getting blood samples and material to Victoria, so and particularly interstate. So we try and make sure that we sort of tee up patients' clinic appointments with a blood test for research, and yeah, that logistics of patient sample handling and transport is a challenge. Consent process and language that's used with patients and families. So when we consent, and making sure I'm explaining what a biobanking is and what the samples will be used for. Data access and centralising, which is a challenge in many research projects. So particularly when patients have had a diagnosis many years back, and sort of tracking down that information can be quite challenging as hospital systems have changed. And then funding as well, so and making the program sustainable. Thanks, Pete. So now I'll hand over to Pete. So once we actually consent a patient, so they're deemed eligible, consent, and then get their sample, it then goes over to the laboratory, and I'll hand over to Pete, who will explain what happens to the samples from there. Thanks, Pete.
Great, thanks, Emily. So once the patient sample comes to us, and it's usually a blood sample or it can be a skin fibroblast sample, we're actually able to create a special type of cell called an induced pluripotent stem cell. So I'll talk a little bit more about this, but these are special cells that allow us to basically grow any muscle that we like. And this is kind of what they look like down the microscope. Occasionally, these cells smile back at us as well, but these are little red dots are the induced pluripotent stem cells that we use in the lab. And so you might be wondering what an induced pluripotent stem cell is. Well, so these are blood and skin cells that have been exposed to different chemicals and different growth factors, which are listed here. And this process was actually awarded the Nobel Prize for Medicine back in 2012. But this is a process where we can turn these blood or skin cells back into a stem cell-like state. And so that's why they're called induced pluripotent stem cells, because we make them in the lab. And the really great thing about stem cells is that they actually can grow into almost any tissue of the body. So these are some examples. So these are hematopoietic progenitor cells. These are our early blood cells, pancreatic beta cells, different types of neurons, adipocytes. But what we're particularly interested in is skeletal muscle as we work on skeletal muscle diseases. So the usual way that you'd have to get muscle is actually from a reasonably invasive muscle biopsy. And these aren't typically collected anymore because we're able to diagnose a lot of the genetic conditions that we are studying by just a simple blood test. And also having to get a biopsy from a healthy patient, a healthy child, is next to impossible. So this really makes this kind of tissue really hard to find, but it is quite important for us. So the benefit of stem cells is that basically they can grow forever, they're immortal, and it gives us a really unlimited source of tissue for us to be able to work with. So once we make these muscle cells in the lab, we can do a lot of different things with them. They're really an important tool. So we can grow them on little flat culture dishes. So this is an example here, and we call them monolayer or 2D cells. We can grow them in more complex 3D muscle organoids, and these are muscle cells that wrap around these two plastic poles. And we're also able to inject these into mice, these muscle cells into mice, and we create like a humanized, a patient mouse. This is work that Paul Grigorovich and Gordon Lynch are doing, and we're not going to really have time to talk about that today. But really the benefits of these cells, or the use of these cells, is quite wide-reaching. They really give us the opportunity to improve the way we can model disease and look at different studies and functions. They can be used to screen different drugs and treatments, and they can look at different pathways to try to correct what's going wrong. So one of the things that we probably think about your muscle is that it's really it's used for walking or for movement. And so what we really want to know is, do the muscle cells that we grow in the lab contract and produce force? So these are what the muscle cells look like down the microscope, and hopefully you can see now we've just given these muscle cells a little bit of electrical stimulus to get them moving. And you can see them contracting here in the dish. So that's really nice. It shows our muscle cells are functional and they can work. And so we're able to do this in 3D. So this is one of those 3D muscle constructs that I showed before. And then we can use really detailed imaging techniques and analysis tools to be able to zoom in and see what's going on within these muscles. So they're quite a powerful tool. So based on the samples that we have in the bank at the moment, we've been able to create a number of different stem cell lines, and they really start and feed into this process of disease modeling down here. And I'm going to hand over to Meagan and Avnika, who are both going to give you some specific examples of how we're using these cells, as well as mice and fish, to be able to do additional screening and testing to be able to start to identify some new therapies.
Thank you. My name is Dr. Meagan McGrath, and I'm a scientist at Monash University. One focus for our lab is a process that functions with inside cells called autophagy. And we are asking how we can use our knowledge of this essential process to improve the way that muscular dystrophies and myopathies are diagnosed and treated. We know that while genetic therapies are viewed with significant hope by patients, families, clinicians, and scientists, they are not yet a treatment for all. Many disorders remain without effective treatments. We need creative new solutions to close this gap, with a particular focus on treatments that are affordable and can be fast-tracked into clinical trial. This is why we focus on autophagy. So what is autophagy? It is essentially the garbage disposal system that operates inside a cell. We know that it's a really important process because it's present in nearly all living organisms, from plants to humans. And the discovery of autophagy was also a Nobel Prize-winning discovery by Yoshinori Asumi, who was awarded the Nobel Prize in 2016. Autophagy essentially removes anything that is damaged or unwanted from within cells. It is essential to keep muscle healthy. When autophagy fails, this garbage accumulates within muscle tissue, causing disease. So how can we use this knowledge in a meaningful way to help patients? We know that there are an increasing number of muscular dystrophies and myopathies where autophagy does not work properly. Diseases such as this could benefit from treatments that target autophagy. This is important because we know that a lot of work is being done to develop new autophagy treatments, because it's also important in more common diseases, including cancer, Alzheimer's and Parkinson's disease, and even with aging. So can we benefit from advances made in this field? We also have the opportunity to repurpose pharmaceuticals already in clinical use to target autophagy. And we can even use dietary-based interventions to target autophagy. However, autophagy is a very complicated process, and we do not yet have good diagnostic tools to identify when this process fails in patients. We are working on changing this, including the development of blood-based tests. We are also using our national network of doctors and scientists to develop a new autophagy testing and therapy pipeline. This begins with the doctors that see patients, and in many cases, patients will undergo genetic testing to identify the cause and type of disease. From here, blood is collected from patients and used to generate a muscle cell model of the patient's disorder, as already discussed to you today in this presentation. And we can use these cell models to do our detective work. And this is because biological processes are very complex and sometimes can be challenging to study when looking at whole tissue. And autophagy, which is a very complex process, requires intensive investigation in muscle cells, further highlighting the importance of us having these cell-based patient models. And as autophagy is such a complex process, it also often requires the use of very powerful microscope systems to study this process and identify what goes wrong when it fails. From there, we can also examine autophagy in patient muscle tissue, and we can also generate mouse models to better understand the disease process and also to begin to test autophagy treatments. Now I'm going to take you through some case studies showing how we have diagnosed autophagy disorders using this type of approach and therefore provided new opportunities for possible treatment. In the first case, we have an example of a disorder called reducing body myopathy, for which genetic diagnosis has been very problematic. Muscle biopsies were taken, and an important feature that was observed in these muscle biopsies was the accumulation of rubbish or debris within muscle cells called protein aggregates. And this gave us the first big clue that autophagy was involved. We then removed these aggregates from the muscle, and we looked at all of the protein components that were present within these aggregates using a technique called mass spectrometry. And what we were able to identify in doing this is that there were a lot of proteins that are required for autophagy that were trapped within these aggregates. And in particular, the major culprit was a protein called FHL1. We then moved across to our muscle cell model to show that FHL1 was responsible for the formation and accumulation of this rubbish or debris linked to autophagy failure. And then ultimately, we were able to cycle back to the patient and undergo genetic testing and identify that the cause of the disease was mutations within the FHL1 gene. Now, with this knowledge in hand, we were able to progress to developing the first mouse model of this disorder, which we can use to start to test autophagy-based treatments. In our next example, we have patients who are diagnosed with a new congenital myopathy due to mutations in a gene called IMPP5K. Nothing was known about how mutations in this gene could cause disease. So this time we made the mouse model first, and again showed that the mouse model could recapitulate the human disease. So here we have an example of the disease muscle in the mouse. And importantly, again, we showed that there was a lot of this rubbish and debris that builds up within the muscle fibers because autophagy is not working properly. And now we also have a model in which we can begin to test autophagy treatments. However, we had to go back at looking at the autophagy process in the cell model. And this is because this was a very complex autophagy defect, and we needed to identify the precise defect so that we could better develop a more targeted therapy that would be effective. And so what I've shown you with this presentation is that it really does take a village of doctors and scientists to advance diagnostics and treatments for patients with muscular dystrophy and myopathy. It also takes a village of research tools, and so often we need to combine the cell-based models that come from patient samples and animal models and also patient tissue to understand disease processes and develop and test treatments. And of course, while we have focused on autophagy in this particular example, our pipeline is adaptable to a whole range of different disease processes, all of which start with the use of patient cell models. Another example of a type of model that's really important in researching muscular dystrophies and myopathies is the zebrafish model, which Avnika will be speaking to you next.
Thanks, Meagan, for introducing the zebrafish model. And so we'll go right into it as to why we're using the zebrafish model as a complementary model to the patient iPSC cells or the mouse model to study neuromuscular disorders. And so this is work led by myself, but also by Professor Peter Curry at the Australian Regenerative Medicine Institute. And so, zebrafish, quite often when I say zebrafish, people are thinking, how can a fish model actually, model a muscle disease? In fact, what's quite remarkable is that zebrafish, the muscle structure of zebrafish is highly conserved to that of humans. And so this is really important. So as we study, disease mechanisms, we can start to, understand the whys and the hows using a model that has very similar muscle structure. But beyond that, zebrafish also have various other unique advantages, and these are the reasons why we really like this model. So, for example, zebrafish offer prolific reproduction. So one male and one female can give hundreds of eggs, and this is really important because these large number of sample size can allow us to more effectively understand if a process is really impaired or not. These embryos are optically transparent, which essentially means that we can see through them. And this is important because we can label our cells of interest or structures of interest and visualize the process happening in real time. And this is exactly what this image is meant to show you. This is a 2-cell staged zebrafish embryo. This is the yolk, and this is one cell, and this is the second cell, and hopefully you can appreciate how transparent it is. And more importantly, these embryos then undergo rapid development. And so this is the real time lapse. It's not a cartoon, it's in real life, where you see this zebrafish embryo developing. And so we started off with 2 cells, and very, very quickly, over 24 hours, this embryo undergoes development such that by 24 hours, we have something that essentially looks like a fish. And so as you'll see in a second, we've got the head forming on this side. We've got the tail forming right there. This structure here, you can see these chevron-shaped structures. These are the somites, and they're primarily composed of muscle. So within 24 hours, we've got something that looks like a fish, and we've got muscle form. It's in terms of phenotypes. I mean, if you think about Duchenne muscular dystrophy, the mouse model of the disease essentially shows pathologies at about 6 weeks. In the fish, we see phenotypes as early as 3 days, and so it's very quick. And so, from a feasibility perspective, we can test therapies or study mechanisms relatively quickly. In addition to that, these embryos are externally fertilized, and why this is important is because we can access that single cell. We can go in and put, DNA of our interest, so transgenesis, or we can go and delete a gene from the fish. And this is just one example whereby we've used transgenesis, so taking a human gene and put that in the fish. So in this case, BAG3P209L is actually it's a mutation in BAG3 that has been shown to cause myofibrillar myopathy in patients. And in fact, in these patients, what you normally see is aggregates forming in their muscle. And in fact, what we've done is taken this human BAG3P209L and we've put that into the fish muscle. And what's quite remarkable is you see these aggregates forming as well. So once again, the pathology we're seeing in the fish is recapitulating what we see in patients as well, allowing us to, identify, disease mechanisms. As we develop these various tools, we can then perform live imaging because these embryos are optically transparent. We can visualize processes happening. And so this is a single cell from a live embryo whereby a protein that's been implicated in spinal muscular atrophy has been labeled in green. And we can then start to visualize, how this protein is moving around our cells. So proteins in our cell are actually quite dynamic, as you can see in this video. And it could be that in certain disease states, how the proteins move are disrupted, and that's what's causing our muscle weakness. So in this case, we can visualize this, we can quantify this, and hopefully better understand what the reasons are for the weakness in muscle health. But also beyond that, we can start to look at non-muscle cells. And so, of course, neuromuscular disorders, as the name suggests, it's also, for example, the nerves that are getting affected. So in this particular model, we've labeled the muscle in blue, and we then have the neurons, and so the nerves are marked in green and red. And hopefully what you can appreciate is that in this particular model, the motor neurons are disrupted. So yes, it's a muscle disease, but the motor neuron biology is also affected. So we can start to visualize and understand how other cell types are affected in our models as well. And finally, also looking at subcellular organelles. And so in our cells, we've got these striated structures. And in this case, in red, this is the sarcoplasmic or the endoplasmic reticulum. And this holds all the calcium that we have in our muscle. And it's really important because it allows the muscle to contract. And in green, we have the mitochondria, which is essentially the powerhouse of the muscle. And so in some disease states, it could be that this calcium release is affected, and that's what's causing muscle weakness. So it could be the mitochondria are not functioning correctly. So once again, using live imaging strategies and using transgenic lines, we can visualize how these various sort of organelles of the cell are disrupted. And finally, of course, the ability to do muscle function assays. And so, a patient goes into clinic and most commonly the clinician will perform what's known as a 6-minute walk test. So determining, how the patient is, the muscle is working in this patient. So in the fish, we've developed some analogous sort of assays that allows us to assess neuromuscular function in these fish. So one such example is this touch-evoked assay. So essentially, we touch the fish on its head, and as a response to that touch, the fish very, very quickly swims away. And so we can measure how quickly it swims away, and this is essentially directly proportional to the neuromuscular function. Additionally, I guess an assay more analogous to the 6-minute walk test is the 10-minute swim test. So in this case, what we do is we put fish, one per well, in this multi-well plate, and we then have a high-speed camera that records the movement of these fish. And so as you can see, the fish spontaneously move. And so over a 10-minute duration, we can record all sorts of parameters such as the distance traveled, the speed traveled, how many times it moves, and so on. And we can try and get an assessment of, how well the fish is moving. And so I guess, you could call it a 10-minute swim test. And so, using a collection of these techniques and many others that I haven't really spoken about today, what we can use the fish model is for improved diagnostics. So to understand if a particular disease variant is really, causing disease, to understand disease mechanisms. So, what goes wrong, or what's, how is muscle wasting occurring, so providing an explanation for this. And finally, keeping in mind that these fish actually live in water. So we can add all sorts of drugs to the water and examine if we can, improve, for example, muscle function and look for improved strategies for therapies from there on. And so finally, I just thought I'd give you a few examples of the sorts of models that we've previously created and how they recapitulate with the patient condition. So here is an image of a model of Duchenne muscular dystrophy. So in these fish, they have a mutation in the gene dystrophin, which is what you see in patients as well. And in this case, we've imaged the whole tail of a 6-day-old fish. In green, we've got the muscle cells themselves, and in red, we've got the membrane of the muscle cell. And so what you can hopefully appreciate is that you've got these holes in the muscle where there's no green. And so in this case, the muscle fibers have fallen apart, they've detached, and that's exactly what happens in Duchenne muscular dystrophy. And you can see how widespread it is. And if we do a muscle function assay on these fish, they do show a reduction in the distance they travel, for example, in a 10-minute swim test. We also have previously generated models of Nemaline myopathy. And so in this case, the gene is TMOD4. And so patients with mutations in TMOD4 show Nemaline myopathy. And so here's an example of wild type. So as I mentioned previously, muscle is beautifully striated. It's actually quite a stunning tissue to look at. And you see these beautiful, like railway track-like pattern. Whereas in these fish, when they don't have TMOD4, you get a loss in that striation. Instead, you get these aggregates forming. So it's exactly what you see in patients with Nemaline myopathy. And in fact, if you use Gamari trichrome, which is quite a common histology stain that's used on patient material, once again, you can see these dense sort of aggregates forming in the cell. Here's another example. So LMOD3, once again, mutations in this cause an Nemaline myopathy. So here is a wild type animal. You can see this beautiful striation pattern. In this case, in these LMOD3 deficient animals, you can see these aggregates forming. So, again, similar to what you see in patients. And finally, here's your mutant in integrin beta-1. In a wild type animal, you can see these beautiful muscle cells, whereas in fish that lack integrin beta-1, they have this impaired, muscle architecture. Once again, very similar to what you see in patients. So I guess, the point of this slide was essentially to tell you that, zebrafish models, when you try and get rid of a gene in a zebrafish, it actually models what you see in patients and beautifully recapitulates these various conditions. And in fact, we have generated various other models as listed in this table, and we are starting to, study disease mechanisms and do diagnostic studies, but also looking for therapies using this fish model for these various conditions. And as we identify, various mechanisms or therapeutic strategies, we then feed back into sort of the wheel that we showed you earlier in the grant. So we've got Peter Houweling, who's been developing iPSC lines for various diseases. So we go back and, tell him, can you test this drug in it? Or can you look at this as a mechanism in your patient cells? But beyond that, also with Christina Mitchell and Meagan McGrath, with their mouse models and with Gordon Lynch and Paul Gregorovic. So combined, collectively, we're starting to talk to each other and really, communicating as to where do we go from, fish into these various other platforms. So that's, coming back to the whole circle, I guess. You heard a little bit about the clinical network and about how all the disease modeling platforms and the therapy screening platforms work together. And I think it's, it's quite amazing to see this circular loop. But I think one thing that I really want to emphasize, we're really, really grateful and excited that, this is the first national program that's really integrating patient advocacy groups such as yourself in this sort of front. And we're super excited by that because I think, the ability to talk to each other, the ability to openly communicate and try and help each other, I think that's the only way that we're really going to be able to make a difference in a field such as in neuromuscular diseases. And so we're really excited that, following this MRFF funding, we've been able to create the first national platform that brings all these clinicians, researchers, and patient advocacy groups such as yourself together on one platform to openly communicate about, what we're doing and where we go from here. So with that, I'd just like to finish up and talk about, I guess, the importance of patient advocacy groups. I first want to say thank you to each one of you. I think it's really important that, you continue to support us and, we acknowledge everything you've done and we're very grateful for that. So I'd like to start by saying thank you to each one of you. But I also wanted to, I guess, talk about how can we help each other more. There's things that you do and things that we do, and I think we need to, how can we work together better? So I think, the first thing is that hopefully in situations like this with the MRFF grant, hopefully this is a first step in closing the gap, by encouraging and promoting partnerships, whether it be between families and patients, but also with clinicians and scientists. So I think that's really important because that's what this sort of opportunities allowed us to do. I think it's really important as you as the patient advocacies that or advocates, you're bringing patient and family voice into our mission. And that's really important. We as scientists, we get so bogged down under a microscope looking at a fish moving. We forget about the bigger picture of our work. And so, you being involved in our research, gives us reason, gives us meaning to our work, and that's really important. And importantly, from our end, outreach and with the patients is really important to improve our success as well. And I think the only other message that I really want to hone back to you is that this is only the beginning. This was the first initiative from the MRFF and a first call out for congenital muscular diseases in 2022. In fact, it was quite a modest amount of funding, 2.5 million over 5 years. And we're hoping that there will be many, many more. And so, if you could use your advocacy and advocate to the government for continued and improved research funding, because, if we as researchers ask for money, they're probably going to say they're all whining about things. But if you as advocates out there go there and ask for more money, I think that will really help. So it's really important that this, yes, this is the first step, but, this continual communication is really important if we want something bigger to come out from this. With that, I would like to finish up. I'd like to thank once again, thank each one of you and all our partner universities and institutes and all our clinicians, researchers, and everyone that's really involved in this project. You've heard from many of us today, and all our email addresses are right here if you'd like to get in touch with us or have any specific questions. But apart from that, yeah, we're very open to addressing any questions that you may have. Thank you.
Got one at the back here.
I hope it's not a silly question, but how is it caused? Nervous system or?
Sorry?
How is this disease caused?
Which one? Sorry. Yes, so which disease were you referring to? Sorry?
I'm not sure about my condition, but I was just diagnosed with a myopathy. I didn't understand it. That's why I'm here today.
Yeah. Unfortunately, I mean, I'm not a clinician, so I won't be able to, tell you that directly. What I do recommend is definitely go speak to your clinician, and they'll be able to advise you better as to, what the genetic background is, and importantly, advise you on, how your disease, or if there's any known mechanisms of how your disease is occurring.
Thank you.
Okay.
Lynn, can you get them to hold it up?
We can have somebody come and talk to you in the break as well and afterwards.
Hi, I was just wondering, you talk about collecting samples from at children's hospitals. Are you only collecting samples from children, or are they adult samples as well?
I'll let Emily answer that. Emily?
Adult samples as well. So how we have predominantly recruited adults is we've actually used advocacy groups to sort of help us bring awareness to the project, and in their email database send out some information on the study, which has actually worked effectively. And then adults and patients and families will contact me, and then we bring them into the Children's Hospital for the blood sample. So yeah, the listed sites on the project and on the ethics are the Children's Hospital, but we can recruit children and adults, and we have been recruiting both.
Are there any other questions from the room?
Hi guys, thank you for that presentation. I was just wondering if you could share, I guess, some of the challenges that you're having in actually recruiting people to give samples for that.
I think, yeah, initially when we got ethics, the clinician clinic was the sort of initial challenge, in that it's sort of making sure that it's the studies on the forefront of their mind and sort of reminding them to let us know of eligible patients. So that, I guess, took a lot of work from my end, whether it be flyers, and, it's a lengthy project to explain and to consent properly. So clinicians that have already have their time slot to talk about a lot of, clinical matters with patients, that sort of adding on a lengthy research consent, it's a bit challenging. So yeah, we sort of just had to make sure that they're aware of the study. The process was to refer patients to me, so then I can have that lengthy conversation with the consent form and the printed information. So it's just sort of reminding clinicians and making them aware of the study. That was probably issue number one. And then we then sort of started using patient advocacy groups to bring more awareness to the study, and then patients were making independent contact that way. And then now probably recruiting has been the challenge. There has been more logistical in getting, patients into the clinic, and we haven't, we're now sort of aiming to start recruiting interstate, and we don't have someone like myself on site at all the other sites. So I'm based in Victoria, but it seems to work a lot better when you've got a dedicated staff member on site, able to, collect the sample, be there to liaise with the nurses, because it's got to be handled in a certain way to get it to the lab. So we're now wanting to start to recruit interstate, and then we're just going to now have to work out how we get, if there's not someone on that other end, in Queensland, for example, how do we then send, who's that going to be, that person sending that sample to Victoria and making sure that it's labelled correctly. So that's will be the next challenge. And I think as well, it's, it's a broad, so I suppose with the ADAPT-CMD, when that group started with the grant, it was sort of congenital muscle disease, and the scientists within that group have a disease type that they're focused on. And then when we set up the National Muscle Disease Bio-databank, we wanted to keep it quite broad, in that we could recruit all the different types of disease types. But of course, there sort of becomes a limit of resources at our end and funding, and unfortunately we can't recruit every single patient. So we just have to be, and sort of if a scientist is of interest to that group, that there's something that they're studying, then we sort of base call outs based on that. So that's how we've sort of worked with our recruitment workflow, if that makes sense.
Thank you. I think we have a couple of online questions. Sarah? Yep, we do. Our first question is from Rachel Spencer, who's the advocacy lead at Muscular Dystrophy Foundation Australia. Her question is, the number of patient samples in the registry bio-databank seem quite low. Assuming you want more referrals, how can MD/MMC support and peak organisations better support the registry and encourage people to participate?
Yeah, I think just to probably make the differentiation. So we've got a registry and then we've also got the biobank. So they're in fact two separate projects. So the registry has been running for a number of years, and that's run by Robin, and that just collects patient information. So patients in the room are likely, and I hope, to be on the registry, so that you get a phone call and there's someone that sits on the phone call that collects quite longitudinal data about how you're going, when your diagnosis was, so on and so forth. So that builds up a really strong base of data, and that's called our registry, our neuromuscular registry. But within that, it couldn't collect samples, so blood samples, muscle samples, skin samples, and that's very valuable material to researchers. So that's why we then set up, or part of the reason why we set up the biobank, because we're then able to set up to collect samples as part of the biobank. And in that, we do also collect some patient information, but of course we sort of don't want to double up with what we're collecting in the registry. So to recruit to a registry is easier because it's, generally a phone call, it's information. We're not having to get a blood test, we're not having to sort of access muscle. So the numbers on the registry, and I can't remember what they're up to, but I think they're in the thousands, and that's been running for a number of years. And in terms of low numbers for the biobank, I think we're actually doing quite well with our numbers. And in terms of peak bodies that can help support, as I said, I've found that recruitment is very effective. So generally organisations such as yourself have a database of patients that you email, so sort of sending out some information on research and asking patients and families if they're interested to participate. That really helps our recruitment. And I think, as Avnika said before, sort of really advocating in terms of funding and making it sustainable. That's something that's really important in advocacy groups. And I think, I'm not a scientist myself. I'm actually a nurse by background, but the scientists love hearing what, impacts patients and how that can help drive their research. So I think that sort of feedback, and even from my point of view in recruiting and how, what information you want from us, I think that's also really helpful. So hopefully that answers your question.
And yeah, just to add, we're very happy as scientists. I mean, we love hearing from you as well. We're very happy to host tours, and if it's just the open communication, the transparency. In the past, we've all worked in our little sort of silos and not been communicating. So I think, if, for example, from a research perspective, if it means, opening our doors to welcoming and showing you what we do, we're very open to do that. So to all the groups out there, if that's something of interest, definitely get in touch as well.
Thank you.
I think there's one last question. We've run out of time for this, but one last question from Sarah Shaw. If you're not on the registry, how do we make that happen? She leads our community liaison group, which is made up of people with lived experience. Wondering if someone could answer that for us.
I'll put my email in the chat, and I don't run the registry myself, but I will pass you on to the person that does run the registry, and I'll send you the website to the registry. So if you're happy, I will get someone from my team to contact you about the registry. I'll put my email in the chat.
Thank you. That's wonderful. Thank you both. That was great. It was really informative, the whole session. So please pass that on to the ADAPT-CMD team and thank them very much. I know that there'll be a lot of discussion. If we have any further questions, we've got some here from online. If we're able to send them through to you, that would be great.
Absolutely.
Thank you.
Thank you for having us here, and more importantly for all the work that you do. We really do appreciate that. So thank you.
Thank you.
Thank you.
Thanks.
Thanks so